Welcome to the deep dive. We're here to really dig into complex topics and pull out the key insights, basically giving you a shortcut to getting informed. Have you ever really paused to think about the hidden architecture inside you, what actually holds us together and makes everything, work? Okay, so let's unpack this today. We're doing a deep dive into tissues, the fundamental building blocks. And our guide is a classic, Histology, a text and atlas, the 8th edition. It's packed with microscopic details.
Absolutely. And understanding tissues, well, it's the foundation. If you want to grasp how the body works, from a single cell right up to an entire organ system, you have to start here. So our aim today is really to give you that shortcut, distilling the vital info about this microscopic world, focusing on how structure relates to function, and importantly, the clinical side too.
Right. So our mission, clarify tissues. We'll look at what they're made of, what they do, where they come from, making the microscopic feel visible and relevant for you. Ready to dive in? Okay, so first things first. The source defines a tissue. How would you put that in simple terms for us? What is a tissue, fundamentally?
Well, basically, a tissue is an aggregate or group of cells organized to perform one or more specific functions. It's a team effort. And the surprising thing, as you hinted, is that, the cell is the basic unit, but it's really the collaboration of cells within these tissues that keeps everything running.
Like an orchestra, maybe individual instruments, but together they make the music.
That's a great analogy. Exactly. The collective effort is key.
So how does that collaboration work microscopically? How do they, stick together and coordinate?
Well, it's pretty sophisticated. They're not just floating near each other. They have specialized anchoring junctions, which are like strong rivets holding cells together, maintaining the tissue structure. And then for communication, they use intercellular gap junctions. These are tiny channels letting small molecules pass directly between cells. It helps them act as one unit.
So they're physically linked and they can talk to each other directly.
Precisely. Plus, they have specific membrane receptors. These let the cells respond to signals, hormones, nerve signals, even physical forces. It all works together so the tissue functions as a unified whole.
Okay, so it's a highly organized communicating team. Now here's what I found really fascinating. The source says, despite all the complexity in our organs, everything boils down to just 4 basic tissue types. What are they?
That really is the simplifying principle. You've got epithelium, connective tissue, muscle tissue, and nerve tissue. That's it. Every organ is built from combinations of these 4.
Just 4. Wow. And how do we tell them apart? What's the basis for classifying them?
It's interesting. Epithelial and connective tissues are mostly defined by their morphology, their shape and structure, what they look like. But muscle and nerve tissues, they're primarily defined by their function, what they do. Muscle contracts, nerve conducts impulses.
So structure versus function, mostly.
Mostly. There are nuances, of course, like myoepithelium. It contracts like muscle. But because of its location and origin, it's actually classified as epithelium. So, there are edge cases, but those 4 categories are the fundamental framework.
Got it. That framework is super helpful. Okay, let's dive into pillar number 1, epithelium. The body's covering and secreting layers. What are its key features?
The 2 big ones are close cell apposition. The cells are packed super tightly together, and it's always found at a free surface.
A free surface meaning, like the outside of the skin, or lining something.
Both, exactly. It covers the body's exterior, lines internal cavities like your chest cavity or abdominal cavity, lines tubes like your digestive tract or blood vessels, and it also forms glands. The cells are always contiguous, side by side, forming sheets, very little space between them. That's crucial for forming a selective barrier.
Okay, tightly packed, lining surfaces. Given they're found everywhere, how do we classify them? How do we make sense of the different types?
It's actually pretty logical. It's based on 2 things. The shape of the cells and how many layers there are.
Shape and layers. Okay.
For shape, you've got squamous, really flat cells, think scales, then cuboidal, sort of cube-shaped, and columnar tall and column-like.
Flat, cube, tall, got it. And layers.
Either simple, just a single layer of cells, often involved in absorption or secretion where things need to pass through easily, or stratified, which means multiple layers stacked up, usually for protection.
Simple for passage, stratified for protection. Makes sense. Can you maybe paint a picture for us? What would a couple of these look like under the scope?
Sure. Imagine looking at simple cuboidal epithelium, maybe lining a kidney tubule or a small duct in the pancreas. You'd see a single neat row of squarish cells. One side faces the open space, the lumen, and the other rests on underlying tissue.
Okay, neat squares in a row. What about columnar?
Right. So simple columnar epithelium, say lining the stomach or gallbladder, These cells are distinctly taller than they are wide. Still a single layer, still with that free surface facing the lumen and a basal surface resting down.
Taller cells, still one layer, and stratified, like in the skin or esophagus.
Exactly. Let's take the esophagus lining. Stratified squamous epithelium. This is where you see the layering clearly. Multiple cell layers stacked up, but only the very top layer, the one facing the food going down, is actually squamous flattened.
So the name comes from the top layer.
Correct. The cells underneath are more rounded or cuboidal, and that bottom layer, the basal layer, often looks like a darker band because the cells are smaller and packed tighter. It's built for resisting abrasion.
That layering makes perfect sense for protection. You mentioned epithelia have other features too on their surfaces.
Oh, absolutely. That free surface can have special modifications linked to function. Things like microvilli, tiny projections that massively increase surface area for absorption, like in the gut. Or cilia, which are like little hairs that beat rhythmically to move things along, like mucus in your airways. There are also stereocilia, which are basically very long microvilli found in places like the inner ear.
So the surface structure really tunes the function.
Precisely. And one more key point. All epithelia, no matter the type, rest on something called the basal lamina. It's a thin, sheet-like layer underneath that acts as an anchor, connecting the epithelium to the connective tissue below. Very important structural support.
Okay, anchoring sheet underneath. Got it. Let's shift gears then to the second pillar, connective tissue, the body's support system. What's the defining feature here, especially compared to epithelium?
The absolute defining feature is the extracellular matrix, or ECM. Unlike epithelium, where cells are jammed together, in connective tissue, the cells are noticeably separated by this matrix material.
So it's all about the space between the cells.
Largely, yes. And the cells actually produce this ECM themselves. The nature of that matrix, whether it's fibrous, gel-like, fluid, or even mineralized, determines the tissue's properties and how we classify it.
And where does this stuff come from originally?
Embryonically, it all arises from the mesoderm, one of those three primary germ layers we talked about earlier.
Okay, so the matrix is key.
Yeah.
What are the main categories? I know there's loose and dense. What's the difference visually?
Good question. If you look at loose connective tissue, maybe just under the epithelium of your skin or lining your gut, you'd see, well, loosely arranged fibers, mostly thin collagen fibers, and quite a few cells scattered around.
What kind of cells?
You'll always find fibroblasts, which are the main cells making and maintaining the matrix fibers. But you also see lots of wandering cells, like immune cells, macrophages, lymphocytes, mast cells ready to react if needed. It's kind of the body's packing material and first line of defense.
Makes sense. And dense connective tissue.
Contrast that with dense connective tissue, like in a tendon or ligament. Here, the collagen fibers are much more numerous, thicker, and packed incredibly tightly, usually aligned in parallel. This gives it immense tensile strength.
So way more fibers, fewer cells.
Exactly. The cells, mostly fibroblasts, are sparse and kind of squeezed between these massive fiber bundles. Strength is the priority here.
Okay, loose versus dense makes sense. But you mentioned specialized types too, where the matrix is really different.
Right, and this really highlights how diverse the ECM can be. Think about bone. Its ECM is hardened, mineralized with calcium and phosphate crystals embedded within collagen fibers. That provides rigid support and protection.
Hard matrix for bone. What else?
Then there's cartilage. Its ECM is more like a firm gel. It's rich in water bound to these complex molecules called proteoglycans, giving it resilience and cushioning. Perfect for joints or your nose and ears.
Firm gel for cartilage. Any others?
And believe it or not, blood is considered a specialized connective tissue. Its cells, red blood cells, white blood cells, platelets are suspended in a fluid ECM called plasma, which is mostly water, but rich in proteins.
Blood. That's surprising. So the matrix can be solid, gel, or even liquid.
Exactly. It just shows that the stuff between the cells is incredibly versatile and really dictates the tissue's function.
Okay, on to pillar 3, muscle tissue. The engine. Its defining feature is the ability to contract. How does that work down at the molecular level?
It's all about two key proteins, actin and myosin. Muscle cells are packed with these proteins, organized into structures called thin filaments, actin, and thick filaments, myosin. Now, actin and myosin are in most cells doing various jobs, but in muscle cells there's just a huge amount, and they're arranged in a highly ordered, often overlapping way. When signaled, these filaments slide past each other, causing the whole cell to shorten or contract. That's the fundamental basis of movement.
Amazing. That sliding filament mechanism generates force. So we have three main types of muscle. Can you describe how they look different under the microscope? Let's start with skeletal muscle.
Okay, skeletal muscle, this is what moves your bones. You see very large, long, cylindrical cells, often called fibers. The most striking feature is the cross striations, these visible stripes or bands running across the fiber. You'll also see multiple nuclei per cell pushed off to the edge, right under the cell membrane.
Big, stripy, multinucleated cells. Got it. What about heart muscle?
Cardiac muscle, only in the heart, obviously. It's also striated, so you see those stripes, but the cells are much smaller than skeletal muscle fibers. They're individual cells, often branched, and they're joined end-to-end
Joined end to end. How?
by specialized junctions called intercalated discs. These appear as irregular dark lines running across the fibers where the cells meet. They're crucial for holding the cells together and allowing the electrical signal for contraction to pass rapidly so the heart beats as one unit.
Branched, striated with those special discs. Okay. And the third type?
Smooth muscle. You find this in the walls of organs like your intestines, uterus, blood vessels. The key difference, it does not have cross striations. The actin and myosin are there, but they aren't arranged in that same highly regular pattern.
No stripes. What do the cells look like?
They're typically spindle-shaped, sort of football-shaped, thicker in the middle and tapering at the ends. Each cell has a single elongated nucleus, usually located right in the center. They contract more slowly than skeletal or cardiac muscle, but can sustain contraction for longer.
Spindle-shaped, single central nucleus. No stripes. Clear distinctions.
And in all types, the way the cells are bundled together, usually in parallel, allows their individual contractions to sum up, producing efficient movement for the organ or the whole body.
Makes sense. Okay, fourth and final pillar. Nerve tissue, the communication network. It receives, transmits, integrates information. The star player is the neuron, right?
Absolutely. Neurons are highly specialized for electrical signaling. They have a main cell body, which contains the nucleus. And extending from that are processes.
Processes, like arms.
Sort of. There's usually one long process called the axon, which is designed to carry electrical impulses away from the cell body, sometimes over huge distances. Think of the nerves going down your leg. Then there are typically multiple dendrites, which are shorter branching processes that receive signals from other neurons and carry them towards the cell body.
Axon sends, dendrites receive. How do they pass the message along?
At specialized junctions called synapses. Usually, when an electrical impulse reaches the end of an axon, it triggers the release of chemical messengers called neuromediators, or neurotransmitters, which then travel across a tiny gap to stimulate or inhibit the next neuron or target cell.
Chemical signaling at the synapse. Okay. But neurons aren't alone, are they? There are supporting cells. What do they do?
They're crucial. In the central nervous system, CNS, the brain and spinal cord, they're called neuroglial cells, or just glia. In the peripheral nervous system, PNS, the nerves outside the CNS, the main ones are Schwann cells and satellite cells.
Glia in the CNS, Schwann and satellite in the PNS. What's their job description?
A whole range of things. They provide structural support, physically separating neurons. Schwann cells and some glia produce the myelin sheath, a fatty insulating layer around many axons that dramatically speeds up nerve impulse conduction. They also act like immune cells, phagocytosing debris if there's injury or infection. And certain glial cells in the CNS help form the blood-brain barrier, controlling what substances can get from the blood into the brain tissue. They're absolutely vital for neuron survival and function.
Wow, so much more than just support. If we looked at, say, a nerve, what would we see?
If you looked at a cross-section of a peripheral nerve, stained appropriately, you'd see bundles of axons. Many would appear as small circles, possibly stained centrally, that's the axon itself, surrounded by a clear or lightly stained ring, that's the place where the fatty myelin sheath was, often dissolved during tissue preparation. These bundles are held together by connective tissue layers.
Okay, bundles of myelinated circles. What about a ganglion, like a cluster of nerve cells?
Right, a nerve ganglion is primarily a collection of neuron cell bodies outside the CNS. So you'd see these large, usually roundish nerve cell bodies, each with a prominent nucleus. And surrounding each cell body, you'd see the small nuclei of the satellite cells, like little moons orbiting a planet. Between these clusters of cell bodies, you'd see bundles of nerve fibers, often unmyelinated in this context. It's quite a different picture from the nerve itself.
Fascinating. Okay, we've covered the four types, but where does all this complexity start? How do these diverse tissues actually form during development? Let's talk histogenesis.
Right, back to the very beginning. In early embryonic development, after fertilization, the embryo forms three primary layers of cells. This happens during a process called gastrulation, creating what's called a trilaminar embryo. These three layers are the ectoderm, outer layer, mesoderm, middle layer, and endoderm, inner layer. And here's the amazing part. Everything in the body, every single tissue and organ, develops from one of these three fundamental layers.
Everything from just 3 starting points, incredible. Can you give us some key examples from each layer? What comes from ectoderm?
Sure, ectoderm gives rise to two main branches. The surface ectoderm forms things like your epidermis, the outer layer of skin, hair, nails, sweat glands, and also the cornea and lens of your eye. The other branch is the neuroectoderm. This forms your entire central nervous system, brain, and spinal cord via the neural tube. And also the neural crest, which gives rise to much of the peripheral nervous system, melanocytes, pigment cells, and some facial bones and cartilage.
Skin nervous system. Okay. What about the middle layer, mesoderm?
And mesoderm is incredibly versatile. It forms basically all connective tissues, bone, cartilage, blood, the loose and dense stuff we talked about. It also forms all three types of muscle: skeletal, cardiac, smooth, the heart itself, blood vessels, the spleen, kidneys, and the gonads, ovaries and testes. A huge range.
Connective tissue, muscle, cardiovascular, kidneys.
Wow.
And the inner layer, endoderm.
Endoderm primarily forms linings. It gives rise to the epithelial lining of your entire digestive tract, except the very beginning and end, and the associated glands like the liver, pancreas, and gallbladder. It also forms the lining of your respiratory system, trachea, bronchi, lungs, and the epithelial parts of glands like the thyroid, parathyroid, and thymus.
Linings and associated glands. That's neat. You mentioned the thyroid and thymus. They start connected but then move?
Yes, it's interesting. Glands like the thyroid, parathyroid, and thymus actually bud off as epithelial outgrowths from the pharynx area early on, but then they detach from their origin and migrate to their final positions in the neck and chest. It shows how dynamic development is.
Development is truly amazing. Now let's connect this knowledge to the real world, to a clinical situation. The source discusses abnormal differentiation and tumors, specifically ovarian teratomas. How does understanding tissues help here?
Teratomas are fascinating, though often problematic, tumors. They arise from pluripotential stem cells, cells that have the potential to differentiate into any cell type. So the teratoma can contain a bizarre mix of tissues derived from all three germ layers, ectoderm, mesoderm, and endoderm. The key is that these tissues are completely disorganized, just a chaotic jumble. They're most often found in the gonads.
And you mentioned ovarian versus testicular. They behave differently.
Very differently, yes. Ovarian teratomas are usually benign. They're often called mature teratomas or dermoid cysts because they contain well-differentiated mature tissues. You might literally find teeth, hair, skin, bits of bone, cartilage, thyroid tissue, even brain tissue inside one.
Teeth and hair inside an ovary.
Strange but true. They're typically found in younger women and often have a normal female 46XX karyotype, possibly arising from an egg cell that started developing on its own.
And testicular teratomas.
They're less common, tend to occur in younger men, and unfortunately they're usually malignant. They often contain less differentiated, more immature tissues, reflecting their more aggressive nature.
Big difference. So, if we were looking at a slice of an ovarian teratoma under the microscope, what would we see based on our tissue knowledge?
Well, at low power, the first impression is chaos. No recognizable organ structure. Just a mess.
A jumble, as you said.
Exactly. But then as you increase magnification, you start recognizing familiar faces, so to speak. You can identify distinct mature tissues scattered randomly.
Like what? Can you give examples?
You might see patches of stratified squamous epithelium looking like skin, maybe with hair follicles. Elsewhere, you could find hyaline cartilage or islands of bone forming. You might see glandular epithelium forming cysts, or even nervous tissue resembling parts of the brain, complete with glial cells.
Wow, maybe even muscle.
Absolutely. You could find bundles of smooth muscle or even striated muscled skeletal muscle fibers in cross-section, maybe even cardiac muscle, showing intercalated discs if you look closely enough. All mixed together, completely out of place anatomically, but microscopically identifiable.
This example really drives it home. Even in this disorganized state, knowing the basic tissue types allows us to identify the components. It underscores why understanding these fundamentals is so critical, both for normal function and when things go wrong.
Precisely. Being able to recognize hyaline cartilage or simple columnar epithelium or nerve tissue, even when it's growing aberrantly in a tumor, is foundational to pathology and diagnosis. It all comes back to these basic building blocks.
What an incredible journey through the body's microscopic world. From tightly packed epithelium to the matrix-rich connective tissues, the contracting muscle, and the communicating nerves. Hopefully, this deep dive has given you a new perspective on the amazing organization within yourself.
And think about this. If just recognizing these tissues, even in the chaos of a teratoma, is so powerful for understanding disease, imagine the depth we gain by appreciating how perfectly they work together, integrated and organized, in a healthy body. It's not just about identifying structures, it's truly about understanding the intricate dance of life itself.
Thank you so much for joining us on this deep dive into histology. We really hope you've gained a fresh appreciation for the complexity humming away inside you. Keep that curiosity going and join us next time for another deep dive into the fascinating world of knowledge.